Okay, we're going to summarize the final stage of protein synthesis. You should have just watched the transcription summary. Now here we are with the translation summary. Translation summary, surprisingly, is going to be a little bit shorter than transcription. So, translation. Translation is the final stage of protein synthesis. This is when you actually are producing your protein. Translation occurs in ribosomes. Ribosomes are found in the cytoplasm of cells. Remember what cytoplasm is? Cyto means cell, and plasm means fluid. So cytoplasm is cell fluid or cell liquid. It's the fluid that fills the cell. Ribosomes are in the cytoplasm. Ribosomes can either be free floating around or they can be attached to the endoplasmic reticulum. So, what do ribosomes do? Ribosomes are the organelles of protein synthesis. A ribosome will attach itself to mRNA and read the codons. When a ribosome reads a codon, it will attract a tRNA molecule. T stands for transfer. tRNAs transfer amino acids to the ribosome. The ribosome then moves down the mRNA molecule, reading every codon. Remember, a codon is 3 letters on mRNA. The ribosome will read every codon, attaching new amino acids to each different codon. So, here we are, translation. Translation takes place on ribosomes, but also in the cytoplasm, because where are ribosomes located? Ribosomes are in the cytoplasm, so translation takes place in the cytoplasm on ribosomes. Here's translation. What is this molecule right here? This molecule is mRNA. How did we get mRNA? We got mRNA from transcription. The mRNA molecule then left the nucleus, where transcription takes place. mRNA molecule left the nucleus into the cytoplasm, and now a ribosome is going to attach to it. So, here we have a ribosome. Here is a tRNA molecule. This is an amino acid. This is an anticodon. Every 3 bases on the mRNA is a codon. So this codon here is AUG. That's the codon on the mRNA. The ribosome will read the codon and then attract the tRNA molecule that has the anticodon. Anticodon. What would pair with A on the codon? U. What would pair with U? A. What would pair with G? C. Think about it. Now, what's going to happen? The ribosome will move down the mRNA molecule. Well, first it will attach amino acids, then it'll move down the mRNA molecule, reading every codon, bringing in a new tRNA, attaching the amino acids together, continuing down, reading each codon, bringing in a tRNA, attaching the amino acids together, moving down until it gets to a stop codon. Stop codons bring in release factors, RF, release factors. This stops translation, the ribosome pops off, and you end up with an amino acid chain, a chain of amino acids, a sequence of amino acids. And what is a chain of amino acids? That's right, a protein. A chain of amino acids is a protein. The purpose of translation is for a ribosome to read the mRNA and then to produce an amino acid sequence based on the mRNA code. And the mRNA code came from what? DNA. Okay, we're going to watch a little illustration of protein synthesis. Well, translation, the translation part of protein synthesis. Translation. Translation is the synthesis the making of a protein. It involves mRNA, the ribosome, and tRNA, and the release factor. Now, you're not going to be assessed on the release factor, okay? Initiation, elongation, and termination, don't worry about those either. So, here we have an mRNA molecule. Eukaryotic mRNA. Why is that important? Well, if you don't have a nucleus, then your transcription process is going to be different. So it's important to say eukaryotic mRNA, because this mRNA is produced in the nucleus through transcription. So here we have an mRNA molecule that's in the cytoplasm now. This mRNA is in the cytoplasm. M in mRNA stands for messenger. Here we are in the cytoplasm. Okay, we're going to talk about some structures of the mRNA. You don't have to know the poly A tail. What you do have to know, though, are these codons. mRNA codons. Every 3 letters on mRNA is an mRNA codon. And there's a bunch of them that go all down the mRNA molecule. You don't have to worry about that either. Each one of these codons codes for a specific amino acid. Don't worry about initiation site. Okay. Here we have a tRNA molecule. Whoops. Okay. tRNA. Notice tRNA, RNA, it's a sequence of RNA nucleotides. Uracils, adenines, guanines. You don't have to worry about the production of that. Don't worry. What you do need to know is the anticodon and the amino acid. Really, the most important thing about the tRNA molecule is that it transfers amino acids. And the anticodon on the tRNA molecule will match with a codon on mRNA. So here we go. Here's your anticodon, UAC. Matches with AUG. The ribosome will read the codon and attract the tRNA with the anticodon. And here's your amino acid. Ribosomes have 2 parts. You don't have to worry about that. You have to know the whole thing is a ribosome. Now, the ribosome will then read the next codon. Here it comes. Okay? Don't worry about the A and P sites. Anticodon matches with a codon. The ribosome reads the codon and brings in a tRNA with a matching anticodon. And the ribosome will attach the amino acids together, and it will get rid of empty tRNAs. So look what's happening. The ribosome moves down the mRNA molecule, reading each codon and bringing in a tRNA with a complementary anticodon and an amino acid attached to it. The ribosome then produces an amino acid chain. Just watch. These jelly beans are amino acids. Elongation is the process of a protein being produced. It's a chain of amino acids getting longer. Elongation. When you're making a chain of amino acids. Okay, so as you can see, the growing amino acid chain is getting longer as the ribosome moves down the mRNA. As the ribosome moves down the mRNA strand, it reads each codon. It reads each codon, bringing in tRNA molecules that have amino acids attached to them. Amino acids then get added to the growing polypeptide. Polypeptide. The peptide bond is the bond that joins amino acids together, and poly means many, so many peptide bonds. It's also another name for a protein, a polypeptide. Okay. Now, the ribosome will continue on until it gets to a stop codon where a release factor is added, and then the ribosome pops off. Termination means to end. And what do we have here? This is a protein molecule. Each of these different colors is an amino acid. The sequence of amino acids is determined by the sequence of codons on the mRNA. And the sequence of codons on the mRNA is determined by DNA, the sequence of nucleotides on the DNA. And that's translation, the last stage of protein synthesis. Ribosome attaches to the mRNA molecule, bringing in different tRNAs with amino acids attached, and then you have a growing amino acid chain. Remember that polypeptide means protein. You add amino acids and you get a chain of amino acids, and a chain of amino acids is a protein. The end. Good job.